Hello guys, I am Shubham from ShubhVed Tutorials. This video is about gene mapping from 3-point cross. In the previous video, we saw what is 2-point cross, that is cross between the two genes and the alleles, okay, or cross between two loci. In this video, 3-point cross means that cross between three loci or three genes, okay, and their allelic forms. So let's get started with the video. So basically, we will see first how is 3-point cross, okay. I will draw your chromosomes, right? Just I will draw here. Okay. Then three three point cross. These are the three genes. Capital A, capital B, capital C. And here small a, small b, small c. Right? Same here. Okay. That's it. So now first cross we will see between AB and AB. Okay. I will change the color. The first cross is between this AB AB. So what we will get? We will get two different forms. Okay. See, capital A, small b, small c, and here small a, capital B, capital C. Right? So how this occurred? Basically, how this occurred? Let's see this clearly in the next slide. Suppose I take here, I will just draw, okay, right? So now cross is between this, right? So what happens? We will write only these things, starting from here and this. So that this becomes capital A, small b, small c, right? And here again, small a, capital B, like this is the row. Got it? This is how we got this. So now next cross is between B and C. Okay, B and C. B and C. So again, what will be here? Here will be capital A, capital B as it is, and small c. Okay, here small a, small b, and capital C. Right? Now in the last case, there is cross between these two as well as these two. Okay, so this is called double crossover. Okay, this is called double crossover. Double crossover. So what we get after this? We get the middle one. Only the middle one changes, right? I will show you clearly. Capital A, small b, and capital C, small a, capital B, small c. Okay. So now you will see how this occurred here. See, A, B, C, small a, capital B, sorry, small, everything. Then crosses between now B and C, second case. Okay, then what will we get? In the first case, we got A, B, and this second. Okay, so capital A, capital B, and small c. And here small a, small b, and capital C. Right? Now double crossover. Double crossover. I will draw here. Right? Now double crossover is there between this as well as this. Right? Double crossover. And what will happen? See, this is easy. Start from here like that. Okay? This is one and this is other. Alright? So it becomes small b will come. Only the middle one changes in the double cross. Right? Middle one changes. Okay? This is how we got all the things. Okay? So this, I hope this is clear to you guys. Now we will see how to solve the examples, solve the problems related to this. See, these are parental offerings, true breed, P1. Okay, then F1. What will be the F1? Capital small. Right? This is F1. F1 will be test cross. Okay, test cross. Right? Test cross. Okay, then the chart is given. See, these are the resulting offerings. Right? So from this, what you have to keep in mind always that the most numbers we see, those are seen in parental, right? Okay, so this majority of the numbers are known as parental, right? They have given in this chart. See, these are parental. Okay, and the less numbers, very less numbers, are known as double crossover. Right? Double crossover and parental. So using this, we can remove the gene order first. Okay, we should remove the gene order first. Gene order. So what are parental here? Right? These are parental and double crossover. They have given capital A, capital B, and small c. Okay, and then small a, small b, capital C. So in the double crossover, we know that middle one changes its changes. Right? Middle one. It is the pathway like we saw here. Okay, in the last case, how the double crossover is middle one is changed. Okay, so we can predict here that this C, this C gene is located in the middle of AB. So the what is the gene order here? A, C, B. Right? This is the gene order. Okay. So now using this, we can find the frequencies, distance, map distance, as well as we can draw the genetic map. Okay. So let's see how to find that. First of all, we will see a single crossover between genes B and C. So we'll see first B and C. Okay, B and C. And how to find? Write the numbers. This. See this. First, as well as double crossover. We will write. And double crossover has to be included in every. See, 5 plus 8 divided by total. Total is given as 1000. Right? So I will write here into 100 directly. Right? So we get 70 divided by 1000 into 100. Right? So we'll cut this. So we get here 7 cm. Okay. So this is the distance between B and C. Now we will see distance of A and C. Okay. A and C. I will just erase this. A and C. Right. So we'll write here 81 plus 85 plus 5 plus 8. Total, so into 100. Okay. So what we get here? 179 divided by 1000 into 100. Right. So what we get? 17.9 cm. So from this map distance also we can understand that B and C are very close to each other and A and C are very apart from each other. So the map can be like we can draw map here starting from here. What is the order? A, C, and B. Right. We know B and C are located very close. So this will be 7. 7 centimorgan and A to C is located, they are far away from each other. So this is a map, genetic map we can draw. Right. So this is how we can find the decombination frequencies. First of all, order, and then distance or gene distance, basically. So what we want to do, we want to just see the parental and the double crossover for gene order. And for gene distance, we want to see the crosses, single cross plus double cross. Okay, divided by total offsprings into 100, right? As we did here. Okay, so suppose B and C is there, then we'll take that number 27 plus 30, and then double crossover number also plus 8 plus 31. Likewise with A and C we did 81 plus 85, and then double crossovers 5 and 8, right? So I think this video helps you. If this video helps you, do let us know in the comment section. Thank you guys. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.